So this is the walkthrough for the Population Ecology Lab. So I'm just going to kind of take you through it a little bit, also let you know where the changes are, because since you're going to be working on this individually online, and this lab was originally written for you to work in groups, I'm going to let you know what changes you're going to need to make to the directions that are in the lab manual. So I've got my electronic copy of the lab pulled up here. And so we're going to walk through taking a look at that. So start just here in the introduction. It's got information just like the other ones with introduction. So this lab is going to cover two different types of sampling: quadrat sampling and mark and recapture sampling. So the first part is the quadrat sampling, and it's got some background information on that. And what I've done for this video is I've highlighted the parts of the lab where there are differences. So if we look here, obviously it says for each group. That's going to be different because you guys are working individually. I forgot to highlight this because you're not actually going to obtain physically a 10-sided die, but you can get electronic dice. And so let me click over here. So if you go out online, and there are lots of these out there, so I didn't pick just one. So you can see I was searching for them. So if you go to, for example, free dice online, but like I said, if you just search for free dice roller or something like that, you can use pretty much any of them. If you want to use free dice online, freeonlinedice.com, you can. What this one will do is it'll ask you how many sides do you want your die to have. And so you just tell it. In this case, we want a 10-sided die. And once you have it set, you just click on the dice and it'll roll the number for you. And so you can just keep clicking it and you can keep using this as your die. So you don't need a physical 10-sided die. I don't expect anybody to have that unless you play like D&D or something. So just find an online dice roller to get those 10-sided dice. Instead of having two, you just need to roll the online one twice. So you're going to toss that 3 times, the set of them 3 times to generate random coordinates. So since you're doing them online, if you use a dice roller like this one, you're going to end up tossing the die 6 times. So because you want to get coordinates on this sampling grid. So if, for example, you're doing your first one, you roll the die and you get a 1, then your first coordinate would be 1, and then roll it again and you get a 5. The second one would be for the letter. So 1, 2, 3, 4, 5. So you would have E1 as one of your sampling plots. And so you want to do that to get 3 quadrats. So you'll need to roll that die 2 times to get each of your squares that you need on the board. So you need to be able to shade 3 of these squares. And we use dice because when we do quadrat sampling, we want that to be as random as possible. So again, you can do that doing it that way. There are also actually dice rollers out here online where it will actually give you 2 dice. So you can select 2 dice, 10-sided, and roll them both at the same time. And then you can get your coordinates that way. So that's, I hope, pretty straightforward. So 1 roll of the dice gets you your number. The second roll of the dice gets you your letter. So A for 1, B for 2, C for 3, and so on. And then once you get your number letter, you just go to that square, shade it in. And you're going to shade in 3 squares somewhere on this quadrat. Okay. Then the next part, as far as the sampling of the population, if we were face to face, we would actually have a physical board, but because if we were face to face, everybody would have their own physical board. So it actually kind of works out for us to do it this way, because this way everybody can play with the same board. So you're going to find the board by going on to iLearn and going to the week that you're in. So if you are online, for example, this week, the week of September 20th to 26th, then you're going to go all the way down to the bottom, just like we have for previous labs when we've been online. And you're going to see this folder that says Documents and Videos for. And so there's all kinds of stuff in here. So there's going to be links up here at the top. You'll see where these come in here in a little bit. Then the thing that you're looking for here is this quadrat. So quadrat 1. This has already been done for number 1, 2, 3, for the directions. So I laid out the grid, I counted out the beads. So there are 200 red dots to represent the adults and 50 blue dots to represent the juveniles. So, I'm sorry, I've got those backwards. So there are red dots for the juveniles, blue dots for the adults. So again, red dots for the juveniles, there's 200 of them, and then 50 dots that are blue for the adults. So that Quadrat file has already done steps 1, 2, and 3 because I've already scattered the beads across the board. So again, number 4, ignore the group part. You're just going to count the individuals that match the squares on your quadrat. So if we go back over to the quadrat here, so if, for example, I rolled 1E, so I would go to 1E and I'm going to count 2 juveniles. So that's what would go in my table 5-1. So if I scroll down here, here's 5-1. So I have my quadrat coordinates in here. It would be E1. And I would say no adults, 2 juveniles, and then I'd go count my next quadrat coordinates. So you're going to do that for all 3 of your quadrats and enter the number of adults and juveniles that you find in them. And then you're going to calculate the average number of individuals for your own 3 quadrats. So round to the nearest 10th, as it says here. So you're going to then calculate the population size using the formula that you see here, and then write the estimated sample size for the adults and juveniles in the last row of table 5-1. So then as far as analyzing the results, I don't have the next document posted, but there'll be a document posted here with sample students in it, because there's no easy way for me to have you guys share your data with each other. So I'm going to put together some sample students who have sampled different coordinates on the quadrat. And so they'll make up the rest of your group. And so you'll just use their data. So plug their data in for adults and juveniles. And so you'll enter their estimated populations for the adults and juveniles for each of the students. So you'll be student 1, and then the sample students will be 2, 3, and 4. And the reason why I'm not just giving you the straight numbers is because I'm going to actually have their quadrat coordinates, because there's going to be actually multiple sample students, because they cannot survey the same quadrat as you. So if any of their quadrats match yours, you cannot use that student. So just go down the list until you find 3 students to put in for yours that do not overlap your quadrats. Then you're going to calculate the percent error, as you can see here, using the percent error formula. So your estimate minus the actual. So depending on who you're calculating, we know the actual population is 200 juveniles. So say your estimate was 170. So 170 minus 200 times 100 divided by 200. That will give you the percent error. Some of these will come out positive, some of them will come out negative, and that's okay, because that's just telling you if it's an overestimate or an underestimate. And so just make sure that as you enter these, that you're getting the positive and negative. So you're going to calculate the percent errors for individual students, and you're going to do it for the whole group. So you and the other 3 students that you get out of the list, you'll calculate their average, the average for all 4 of you, and your percent error for each group. Then there's some questions to answer about this section, and then you don't need to worry about the cleanup. You just need to get rid of the file. Procedure 5.2, you'll see I have all of this highlighted. In fact, I gave up highlighting it because you're not going to do procedure 5.2 as it is in your lab manual. All right, you're going to do procedure 5.2 as it is in the document that is here in the folder. So you'll see here there's a file called Lab 3 Ecological Sampling Techniques, Mice in the Forest Ecosystem at Primer. So we usually get the chance to go down and visit Primer, but because of what's going on this semester, we can't travel to Primer. You may or may not be aware that Capitol has a large outdoor learning center, which is really it's a cool place. It has lots of different habitats on it. And one of the things that's being done down there is research on white-footed mice. Dr. Christine Anderson does that research, and so she has actually provided us with her data from last year, 2019, so that we can use it to do mark and recapture with real data, rather than just me trying to figure out how, me having you try and do mark and recapture with, little dots on the screen. So you've got here a document. It's got background information on how mark and recapture is done. It's got information on how to do the calculations. So you've got information in your book, but you also have that same information in a little more depth with examples here in the document. It's also going to tell you some information about this method. So this is a really easy, straightforward calculation compared to other ones, but it's going to talk to you, and I would like you to read through what the assumptions are for this calculation, because there are some downsides to it, and I'm going to ask you some questions about that. The other thing that's here is the other calculation. I'm not going to ask you to calculate this. I've actually calculated the Schnabel Index for you, because it's actually a little tricky if there's not somebody there to kind of help you figure out how this is working. So you can see an example here of kind of how it works. And so then there's some information here about Primer and the research that's going on there. You can see the layout of the Primer site. And you can see we've got lots of different areas. We've got fields, wildflower meadow, a grass area, woodland area, a prairie ecosystem. It is right next to the Hocking River, which is kind of nice. There is a wetland region in here. So like I said, huge variety of different habitats. There are little streams and so forth that run through the woodlands. It is right off of 33, so if depending on where you are in this system, you may actually be able to hear the freeway, which is interesting. So as far as field collection, Dr. Anderson was nice enough to take some videos so you can see how this works. So the 4 videos that are on iLearn, when you open this folder, those are of Dr. Anderson showing you how the traps are set and also giving you an idea of what the ecosystems look like that they are doing the research in. So we've got the forest ecosystem. She talks a little bit about that. The fence row ecosystem. And in both of these videos, she gives you some information about what these white-footed mice are eating, but also how they do the trap baiting and so forth. And then how they actually handle the mice. You can see the little guy right there, and what kind of data they collect on it. And so I want you to watch those 4 videos and then answer the questions that are here about what's going on in those videos, what they're doing as far as the research goes. And then the other file that's there on iLearn, let me flip back over. You'll see there is an Excel file here as well. You'll want to pop that Excel file open as well. That's where the data is that Dr. Anderson sent us. So this is the data about the different mice that they collected in 2019. And so you're going to use this information to calculate the population using this formula. So when you open this, it may open to the forest tab, it may open to the ag tab, it may actually even open to the instructions tab. The instructions tab just tells you to use the first 2 time periods to estimate the population. So let me show you what that means. So in her data, you can see up here, the first date that they sampled in 2019 was 5/11. The second date they sampled was 6/5, and then 6/12, 7/11, 7/18, 7/25. Okay. So what you want to do to use this formula is you want to only use the first 2 dates. So you want to look at the mice that were captured on 5/11 and the mice that were captured on 6/5. So those are the only ones that you want to look at. And what you're looking at as far as the formula is looking for the population size based on marked individuals that were released in that first time, the number of individuals captured in the second sample, and the number of animals that were recaptured. Okay, so we want to look first to see how many marked individuals were released the first time. And so you want to take a look and see, okay, so the first date's 5/11, and you're looking for the data here for recapture, because they may have recaptured somebody from last year. So if it says no, that means it's not a recapture. That's a new mouse, and it gets marked on that date. So it gets a little ear tag that you'll see in the video. And so if it says no for recap, that means it's now a marked mouse for that date. So these guys on 5/11, those are M. Those are marked mice. Then to get your other data numbers, you're going to go to 6/5 and take a look again at what's in this recap section, because you're looking to see who got recaptured and how many total mice got recaptured at that time. And so you can see how many were captured and how many got recaptured. And so that gives you the numbers for your formula. Okay, and you'll do the same thing over here for the forest. So for the forest or the wooded area, the first date is 6/5. So that's the one that you're going to use, the first date. And then the second date is going to be 6/12. So those will be the 2 dates you use the data from. If you have questions about the calculations, obviously let me know. While I was in there, I was going to show you, like I mentioned, I went ahead and calculated the Schnabel. So that way you can kind of compare them. And so I've got the calculations set up in a table here, and all calculated out through using Excel calculations. And then here is the population number. Schnabel comes out as a decimal, so we round. And so you'll be able to see that in both of the Excel tables if you scroll down. So if you go down, you'll see I've calculated that, so you can enter that in. And then there's a couple of questions here to answer as far as the information from this section of the lab. Then if we go back over to the lab in your notebook, in your lab manual, like I said, you're just going to skip 5.2 completely because that Word document we just looked at takes place of the 5.2, since we've got that data and the videos from Primer that we can use. So then you're going to go ahead and you're going to do 5.3. So 5.3 is looking at population dynamics of elk, of Tool elk in one of the parks here in the United States. So you can see there's population data here. So you're going to create a graph. You can create that graph just by hand. It doesn't have to be exact. You're basically just trying to get the trend for the graph. And then there's some other questions to answer about the population trends and so forth that are going on here. And then I do want you to complete, you'll see this listed in the box, the drop box. I do want you to complete the post-lab and pre-lab questions both, because they're good review questions for the material. So the post-lab questions are going to talk about things from both the mark recapture and the Quadrat method. I didn't start at the scroll back up, the pre-lab questions, but those pre-lab questions are the first couple of pages. So if we go back up here, see, they are the first couple of pages. Those pre-lab questions you should be able to answer by reading through the information that's in the lab. So you don't have to do them first. You can do the lab and then go back and answer the pre-lab questions. So that is the walkthrough for the lab. Really, if that's, I mean, it's fairly straightforward once you get working into it. I can't think of anything else. If you do have questions, run into problems, let me know. This is going to be due a week after your lab session. So those due dates are posted up in the Dropbox for the different lab groups. So, and it's also posted, well, no, it's not posted anywhere else. It's posted in the Dropbox. So if you are in lab group A, that would make your lab due the week of September 28th. If you are in lab group B, that would make your lab due the week of October 5th. It is due on your normal lab day in the Dropbox of those weeks. And again, if you have any questions, run into any problems, let me know. That's why I'm giving you that extra week between when you're scheduled to do the lab online and when it's due. So yeah,